Hello friends, I am Shubhangi Rohatgi, PGT Biology from KV1 AFS Indian, and I welcome you all to session 2 of chapter 11 of class 11th, Transport in Plants. In the last session, we had studied about diffusion. Can you tell me what is diffusion? Yes, it is the random movement of particles from the region of their high concentration to low concentration. It happens because of their kinetic energy, and it does not require energy in the form of ATP from the cell. Then we studied the facilitated diffusion. How is facilitated diffusion different from the normal simple diffusion? Well, here the semi-permeable membrane and the carrier proteins are also involved. So, facilitated diffusion is the one where diffusion takes place along the concentration gradient, but help of carrier proteins is taken, so that the molecules which were otherwise unable to pass through the membrane can now easily pass the membrane with the help of carrier proteins. Then we also discussed a little bit about the active transport. What is active transport? It is the transport of materials against their concentration gradient, that is from the region of their low concentration to high concentration. And of course, since this is against the concentration gradient, that means some work is to be done to achieve it, and for that matter energy in the form of ATP is required in active transport. So, this was about the transports. Then we also studied about water potential. What is water potential? It is the measure of free energy of water molecules in a system. Water potential depends upon the solute potential and the pressure potential. Solute potential happens because of presence of solutes in the system, and they lower down the free energy of the water. That is why whenever there is a solution, the water potential is lowered. However, pressure on the other hand increases the water potential of a system. So, pressure potential increases the water potential, but solute potential, it decreases the water potential. Now, in the last part of the class, I had given you some questions also. I hope you have tried to solve them. Let us try to solve them here. First question was, the osmotic potential and pressure potential of three cells, namely A, B and C, located in different parts of an actively transpiring plants are given below. So, these are the three cells and their osmotic potentials and pressure potentials are given here, and you have to identify which of them is the root cell, the root hair, which is root cortical cell, and which is the mesophyll cell. So, as we had discussed in the last class also, in order to solve questions like these, what we should do? We should calculate the water potential of each cell. So, let us begin by calculating the water potential. So, for cell A, where osmotic potential I will first make a table, and pressure potential I can write it like this with the help of symbol only, and we know that sum of these is equal to the total potential. Why do we need to calculate the total water potential? Because then only we will get to know the direction of the flow of water, because we know that water flows from the region of its high water potential to low water potential. So, for cell A, the osmotic potential is given to be negative 0.87, while the pressure potential is positive 0.044. When we add them, we get a negative value of 0.43. Then for the next case, for the cell B, osmotic potential is negative minus 0.92 and pressure potential is positive 0.34. The total water potential will come to be 0.52. Similarly, in the last case, cell C, the osmotic potential is minus 0.68 and pressure potential is 0.27. When we add this, we get that the psi total, that is the water potential, is minus 0.41. So, in this way, in the three systems, we can see that these cells, cell C, have the highest water potential and these have the lowest. Now, the water moves from high water potential to low water potential. We also know that in plants the water moves from roots to leaves. That means this value must be of root hair cell, the highest value of the three. When we are comparing these three, of the three, the highest value of water potential should be of the root hair cell, while the lowest should be that of the leaf, and the one which is in the middle, that should be the cortical cell. So, this should be the answer, that C with the highest water potential is the root hair cell. B with the lowest should be the mesophyll. That is why the transport of water will occur from roots to mesophyll, and the cortical cells should have a water potential somewhere in between the two. Okay. So, let us see which option satisfies our answer. It is the first option. C is the root hair, A is the cortical cell and B is the mesophyll cell. Second question, which of the following denotes the water potential of the mesophyll cell in wilted condition? Now, in wilted condition, all the water has moved out from the cells. Okay. So, what should be the water potential of a cell where all the water has moved out? The answer is that it should be equal to the value of its osmotic potential, that is the solute potential. Now, then this was a match the column question, where you were to match the values with their proper sign, negative or positive. So, water potential of 10% salt solution, pressure potential in a normal cell, pressure potential in a plasmolyzed cell, or metric potential on the surface of the wood. These were the options, and let us see. This way we cannot really know the answer. So, let us see the answer. Obviously, the water potential of 10% salt solution will be negative. Why? Because addition of any kind of solute lowers down the water potential. The water potential of pure water is 0. So, anything lower than this will be a negative value. That is why the solute potential of a solution or water potential of a solution with 10% salt will be negative only. Now, pressure potential in a normal cell, as we had discussed earlier also, that pressure increases the water potential and it has a positive value. So, pressure potential will have a positive value. Well, in a plasmolyzed cell, there is no turgor pressure. So, the pressure potential value will be 0 here. And finally, metric potential we had discussed in the last class, that metric potential has a negative value. So, on the surface of the wood, the metric potential will have a negative value. Let us move on to the next question. Water will be absorbed by the root hair when you have to choose one of the correct options. Concentration of salts in the soil is high, or concentration of solutes in the cell sap is high, or plant is rapidly respiring, that is, it is metabolically very active, or last option, they are separated from soil by a permeable membrane. What do you think the answer is? Yes, the answer is when the concentration of salts in the soil is high. How this happens we will see in a while. Let us move on to the last question. There was this assertion that potassium ion accumulation found in Nightella plants depends on the respiratory activity. Reason assigned to this assertion was absorption of all ions in the plants completely depends on the usage of metabolic activity, and your options were that both these are true and reason is the correct explanation of assertion, or both are true but reason is not the correct explanation of A. Next option was assertion is true but reason is false, or assertion is false but reason is true. The answer is that assertion is true. Potassium ion accumulation does occur because of the respiratory activity. It determines how much ions will be accumulated, but the reason is wrong. Not all the absorption of all ions in plants completely depends on the usage of metabolic activity. It is not true. This was about the last class. Let us move on to today's topic, that is the osmosis. You must have studied about osmosis in junior classes also. You must be knowing that osmosis is also a special case of diffusion. It is the spontaneous net movement of solvent molecules through a semi-permeable membrane. Now, the point to be noted here is that osmosis involves the movement of solvent molecules. Up till now, when we were talking about diffusion, we were talking about the movement of solute particles, but now we are talking about the movement of solvent particles. So, where do these solvent particles move? They move into a region of high solute concentration in the direction that tends to equalize the solute concentration of the two sides. Let us see how this happens. Suppose this is a system containing lot of water, and here a semi-permeable membrane has been placed. Now, the semi-permeable membrane, what is its property? It allows movement of some particles but not all. So, it will restrict the movement of some particles and allow some others to move. Suppose these are the water molecules. I will represent them by dots. Initially, on both sides there is pure water, and since the membrane is semi-permeable, let us suppose that it allows the passage of water molecules from one side to the other. Now, what will happen if some solute particles are added to one side? These solute particles, being big enough, are not able to pass through the membrane. What will happen? Now, please see that though the amount of water molecules on both sides is still the same, but when we talk about concentration, we will say that this solution is concentrated in terms of solutes. Similarly, this solution is concentrated in terms of water. So, this solution is relatively more dilute in terms of solvent or the water than this one present here. Please follow with me that when we add solutes to one side, what happens? The solution becomes diluted in terms of number of quantity of water molecules present here. So, in order to equalize, what should happen? The number of water particles should increase on this side. That is why the water begins to move from the region of its high concentration to the region of its low concentration. So, the water will move towards the side where solute is present, and this is happening because of osmosis. If we want to see in the terms of water potential, let us see what is happening here. Because of presence of the solute particles here, the water potential here has come lower. That means it has decreased. But since there was no solute present here, the water potential is high here. And we have learnt that water moves from the region of its high water potential to low water potential. So, because of osmosis, in such a system where two solutions are separated by a semi-permeable membrane, the water will move from the region of its high water potential to low water potential. Let us see this through an activity also. What we are going to do is that we will take a tube and on one end of it we have fixed a cellophane paper, and now we will fill it with a dense highly concentrated sugar solution. Why we are using the cellophane paper? Well, because it acts like a semi-permeable membrane. You can also use a dialysis tubing, and in nature you can obtain semi-permeable membrane from the eggs also. You remove the upper hard covering of the egg by dissolving it in the acids, and then you will get the inner semi-permeable membrane of the egg. So, now for sake of simplicity and it's easier also, we are using the cellophane paper here, and what we are going to do, we will add some sugar solution into it. Now, this sugar solution is highly concentrated, and in order to make it visible, I have added some food coloring to it also. So, can you see the red sugar solution here? Now, we will also mark its level. So, I am marking the level of the sugar solution here with this green pen. This is important because we need to remember what was the initial level of the sugar solution. So, we have put a mark here. You can see that with the green pen I have put a mark on the level of the sugar solution. The sugar solution is red, appearing red because of presence of a food coloring also. It is not going to interfere with our experiment. Now, what will I do? I will take a beaker with some pure water. This is distilled water in this beaker, and I will put this concentrated sugar solution separated by a semi-permeable membrane into the beaker of pure water. Okay. So, what is our system here? I will repeat again because you need to remember it, that this tube contains dense concentrated sugar solution, and it is separated from rest of the pure water by only a semi-permeable membrane. We will leave it for a while, and after some time we will see what happens. What do you presume will happen? Let me draw a diagram of the system that we have just arranged. So, we have a beaker. In the beaker there is pure water. I am drawing this solution diagram in order to help you remember what was where. So, this was pure water, and we had taken a test tube. On one end of the test tube was a semi-permeable membrane tied to it, and then we filled the test tube with a sugar solution, and we have kept this experimental setup for the observation. We will return to it in a while. What do you think meanwhile will happen? Which should be the direction of flow of water? We have studied just now that because of osmosis, when water is separated in a system by a semi-permeable membrane in such a manner that on one side there is a dense solution, on the other side there is pure water, there is movement of water. Can you predict the direction of movement of this water in this system? Think in the terms of water potential. We will come to the solution later. Let us study about osmosis a little bit more while we are waiting for the experiment to be completed. So, moving on, talking about osmosis, it is a physical process in which any solvent moves across a semi-permeable membrane. The membrane of course is permeable to the solvent, but not to the solute, separating the two solutions of different concentrations. What will happen if the membrane is permeable to solute also? Then osmosis will not take place. The solute molecules will simply diffuse from the region of their high concentration to the region of their low concentration. So, there will be no question of osmosis if the membrane is permeable to the solute particles as well. Osmosis will occur only when the membrane is permeable only to the solvent particles and not to the solute particles. So, osmosis, it can be made to do work. In biological systems, the solvent is typically water, but osmosis can occur in other liquids also, supercritical liquids and even in gases. Osmosis depends upon the pressure gradient, concentration gradient, and so we have the system. This is a diagram that is also given in NCERT, and in this system there are two chambers A and B, each separated by a semi-permeable membrane, and the blue color represents the water and solute particles have been shown with little spheres. Obviously, we can see that the concentration of solute particles is more on the B side and lesser on the A side. Now, let us see the questions that are present. Solution of which chamber has lower water potential? Well, how can we solve that? As we add the solutes, the water potential lowers. The more the amount of solute, the lesser will be the water potential. So, in this case, the water potential will be lower in chamber B. Where the solute potential is lower? Solute potential again because of the presence of solutes. So, where the more solutes are present, the solute potential will be higher, but the solute potential will be lower where the amount of solutes is less. So, it will be lesser in chamber A. In which direction will osmosis occur? Now, again, as we had discussed earlier also, the water potential is lower in B and higher in A. So, the water will move from A to B. Last question, at equilibrium, which chamber will have lower water potential? Now, when we are talking about equilibrium, equilibrium means there is no net flow, and equilibrium means that the things have been made to be even. So, obviously, both will have equal water potential at equilibrium. Moving on about osmosis. Osmosis is very important for the plants. It provides the primary means by which water is transported into and out of the cells. The turgor pressure of a cell is largely maintained by osmosis. When a cell is submerged in water, the water molecules pass through the cell membrane of an area of low solute concentration to high solute concentration. Now, please remember that we are talking about solute concentration, but the movement that is happening is of the water molecules or the solvent molecules. So, for example, when a cell is submerged in salt water, the water molecules move out of the cell. If a cell is submerged in fresh water, water molecules move into the cell. When the membrane has a volume of pure water on both sides, what will happen? Water molecules will continue to pass in and pass out in each direction at exactly the same rate. So, as a result, there is no net flow of water through the membrane. Talking about the osmotic pressure. Osmotic pressure is defined as external pressure required to be applied so that there is no net movement of solvent across the membrane. How this happens? See, when we apply pressure to a system, because of that pressure, the osmosis can be made to be stopped, and the amount of pressure that is required to stop the osmosis, that is to stop the net movement of solvent across the membrane, that pressure is known as the osmotic pressure. Now, osmotic pressure is a colligative property. What do we mean by colligative property? It means that the osmotic pressure depends upon the molar concentration of solute, but not on its identity. Whatever may be the solute, but the osmotic pressure does not depend upon which solute is this, what is its charge, what is its shape or size, but it depends only upon its molar concentration. So, in this diagram from NCERT, let us study the osmosis. So, this setup is actually similar to one which we have set up in our class today. So, there is this thistle funnel and it is filled with sucrose solution. It has been kept inverted in a beaker containing pure water, and at one end of thistle funnel a semi permeable membrane has been tied so that osmosis can take place. And because of the presence of solute and what we have been discussing up till now, water will move into the thistle funnel, and we will see that after some time what will happen, the amount of water will increase in the thistle funnel. Now, if some pressure is applied at any point here, what will happen? The osmosis will stop, and the amount of pressure that is required to stop this osmosis will be known as the osmotic pressure. So, in the similar experiment that we have done today, we will see what happens. So, let us come back to our experiment. We had a beaker and sugar solution. Let us see what has happened. So, only some time has passed and we can see that the amount of water has already increased in this test tube. It is well above the green mark. Can you see it here? The level of water is well above the green mark which we had put earlier. Let me mark the new level here for you. About this much water has entered this tube because of osmosis. Moving back, what must have happened? That since the water was pure here, it means its water potential was 0. And here, since the sugar solution was there, so water potential was highly negative because our solution was very concentrated. So, the water potential of this solution in this test tube was highly negative. As a result, this came to be the lower water potential, this came to be the higher water potential, and this was the lower. We know that water moves from high potential to low potential. That is why the water entered this test tube here, and we could see that the level of water rose in this test tube. This is our thistle funnel experiment that we can perform in our class also. Now, this was only for an artificial membrane, it was not a natural membrane. What about the natural cells, the plant cells? What happens to them when we carry out osmosis? Can we carry out osmosis actually? Okay, let us try to see osmosis. What am I going to do today is cut some fresh fruit for you. Why? Because obviously these will give us the fresh cells. So, we have a lot of fresh cells now, the plant cells, and we will try to see the effect of osmosis. Now, in earlier experiment we had taken a sugar solution. Now, we will use the salt solution. So, we are using highly concentrated salt solution here. I am not using water here, I am just dabbing the plant cells with a lot of salt. Why? Because I do not want you to think that water has come from the salt. Now, have you ever seen this happening, that whenever you prepare a nice plate of salad and put a lot of salt on it, and then if you served it after a while, the salad all became soggy. From where did that water come? The water that has come from the plant cells, and because of osmosis. Since you sprinkled a lot of salt on the salad, the concentration became so high that the outside of the environment of the plant cells, it became hypertonic, and the water was sucked out by the salt from the plant cells. So, let us wait for a while and see what happens when a lot of salt has been dabbed on these plant cells. Meanwhile, I have done something for you. These are the potato cubes, and they were placed in a concentrated sugar solution overnight. So, you cannot actually tell me only by looking at them, but when I give you something to compare with, you can see the difference in the size. Now, can you see the difference in the size of the two? These potatoes were placed in normal pure water overnight, and these have been placed in a dense concentrated solution overnight. But let us not only presume that the size has changed. I have got some photographs for you also. I measured a cube before putting it inside the solution. So, the measurement of longest side was 1.5 centimeters, and this is the real picture. And when I measured it again in the morning after keeping it in the strong sugar solution overnight, I could see a difference, and now the longest side measured only 1.4 centimeter. So, this was the earlier picture before osmosis, and this is after osmosis. So, what has happened? The water has been drained out from the plant cells because of presence of concentrated solution outside the cell environment. This is how osmosis takes place, and otherwise also there are so many examples where we can see the osmosis happening. Think of anything that you need to dehydrate and you use salts or sugar solutions for making them dehydrated. Have you ever seen a mango pickle? How dehydrated, shriveled is it? But a ripe mango or even an unripe one, but the one which is fresh, is not that shriveled. Now think about the process by which pickles are made. Pickles are made using lots and lots of salts, and what do these salts do? They drain out the water. Similarly, in earlier times, the meats were cured by putting them in brine solution. Brine solution is a salt solution, and whenever you put something in brine solution, it will eventually be dehydrated. Concentrated sugar syrups are also used for dehydrating fruits and making them jams and all. But jams, curing of meat and pickles, why do these things happen? I mean, why are these things done? Why do we make pickles, jams and cure the meat? Yes, in order to prolong their life, and by meaning of prolonging their life is that we need to protect them from bacterial and fungal attacks. And when we put a lot of salt or sugar around their systems, what happens? Any bacteria or any fungus that is trying to attack that food item will be severely dehydrated and killed. That is why properly cured meats or pickles or jams can be made to last for a lot of time without being attacked by bacteria or fungi. The presence of salts or sugar, it kills them. So, this was about osmosis, and yes, I will show you that. I hope you will be able to see that a lot of water has started coming out of your freshly cut fruits. Can you see? Yes, if this is kept for a long time, more water will be seen coming out. So, this was another example of osmosis taking place. Moving on, plasmolysis. Now, a solution can be of three types depending upon the concentration of the solute. It can be hypotonic, isotonic or hypertonic. As the name suggests, when the solute concentration is less or the osmotic pressure is also less, then the solution is said to be hypotonic. When these quantities are equal outside the solution and inside, then the solution is isotonic to the system, and hypertonic solution is one which has more solute concentration or more osmotic pressure than the system that we are talking about. The system generally is the cell. So, with reference to cell, a hypertonic solution will be one which has more solute concentration than the cell itself. Okay, and hypotonic solution will be one which has lesser amount of solutes as compared to the cell that we are talking about. What happens when cells are put in these different solutions? For an animal cell where cell wall is absent, the changes are drastic. If an animal cell, suppose red blood cells kept in hypotonic solution, water will move in because of osmosis and it will be burst. In an isotonic solution, the net movement of water is not there. So, water can keep on coming in and moving out, but there is no net movement of water molecules. However, in a hypertonic solution, most of the water is drained out because of osmosis and the cell is shriveled. The effects are also there on plant cells, but they are not that drastic because of the presence of cell wall. The plant cells, they can bear to be in hypotonic, isotonic and hypertonic solutions much more than the animal cells because of presence of the cell wall. Let us see the effect of these solutions on the plant cells. What happens when a plant cell is put in a hypotonic solution? Water diffuses into the cell. This we have been discussing. In a hypertonic solution, when the solute concentration is more outside, the water will move out, but when the solute concentration is less inside, the water will move in. So, because of movement of water into the cell, the turgor pressure builds up and the pressure potential will obviously increase. Turgor pressure is very important for the plants because it is responsible for cell extension and enlargement. So, this is the diagram of a turgid cell. It has been, a cell was put in a hypotonic solution, water moved in and the cell became turgid. Suppose this is a normal cell. This is the cell wall, cell membrane, and a vacuole. When it was put in a hypotonic solution, the water will move into the cell, and now the plasma membrane will start putting pressure, exerting pressure on the cell walls. The cell walls do not break or burst under this pressure, but they bulge out or the cell becomes turgid. This is how a comparatively more turgid cell should look like after being placed in the hypotonic solution for quite some time. If a plant cell is kept in an isotonic solution, there will be no net flow of water, and when water flows into the cell and out of the cell in an equilibrium, then the cells are said to be flaccid. When there is no net movement of water molecules, then the cells are said to be flaccid. That is, the plasma membrane is not pressed tightly against the cell wall, and therefore, the cell is neither swollen or nor plasmolyzed. Lastly, this is the diagram of a flaccid cell placed in an isotonic solution. There is no net flow of water. These arrows depict that water can come out or go in easily and freely. Finally, what will happen when a cell is placed in a hypertonic solution? Plasmolysis will occur. What is plasmolysis? Plasmolysis is moving out of water from the plant cell when the cell is put in a hypertonic solution because of osmosis, and now when the water has moved out, the cell cannot shrink because of the presence of cell wall, but the protoplasm, it gets shrunken. So, in a plasmolyzed cell, we can clearly see the protoplasm that has been shrunken and the water that has drained out from the cell because of the presence of hypertonic solution outside. So, in a hypertonic solution, water will move out. Remember, the animal cell would have become shriveled or shrunken, but since there is cell wall in the plant cells, they do not shrink. The water moves out from the cytoplasm and then from the vacuole also, since the cell wall cannot shrink, but the protoplasm can. So, the protoplasm shrinks away, and this is how a cell looks like when it is plasmolyzed. So, the solution here must be hypertonic, that is why the water has been drained out. Let us have a look at the plasmolyzed cell. Now, there is one question. Suppose this was the cell again and it was now placed in a hypertonic solution. Let me draw it again for you. This was the cell with a large vacuole that occupies quite a lot of space in the plant cell. Suppose this is a normal flaccid cell and it was put in a hypertonic solution with a lot of solutes, that is the solute concentration is high. What will happen? The water will start moving out. After a while, the cell will look something like this. There will be no change in the size of the cell because the cell wall stays where it was, but inside there will be a lot of change. The plasma membrane will shrink. It is somehow attached to near cell wall at some points, but largely it gets shrunk. The vacuole is also very small now. So, my question is that since the water has moved out, what is present here? Are these places empty? Is air present here? What is present here? Earlier we say that there was cytoplasm and the water and the organelles. Now the water has drained out and the cytoplasm, the protoplasm, it has shrunken. So, what is present here at the places? Of course, it is a hypertonic solution which is present here now. Look what happens. After plasmolysis, the gap between the cell wall and the cell membrane in a plant cell is filled with a hypertonic solution. From where has this hypertonic solution come? This is because as the solution surrounding the cells is hypertonic, exosmosis has taken place. Exosmosis means water has moved out and the space between the cell wall and cytoplasm is filled with solutes. So, a lot of solutes are present here, and as most of the water drains away, the concentration inside the cells becomes more hypertonic. So, earlier in the original flaccid cell, there must be some amount of solutes also, right? The solution was hypertonic, okay, but the cell must be having its own solutes also. Now, these solutes remained behind while the water moved out. As a result, the places, they are now occupied by the solute and they are highly hypertonic. Okay. Moving on. So, this is a microscopic image from a cell before plasmolysis and after plasmolysis. You must have done practicals in your classes performing plasmolysis. The last topic for today is imbibition. Imbibition is a special type of diffusion. Water is absorbed by solids collides, causing an enormous increase in volume. An example is absorption of water by seeds and dry wood. So, what happens in imbibition? If it were not for the pressure due to imbibition, seedlings would never have been able to emerge out of the soil into the open. They probably would not have been able to establish. You all must have seen raisins. This is how they look like. They are highly dried and super concentrated, and before putting them in puddings and all, what mama does? Mama soaks them overnight. Why? So that they come to look somewhat like these. Now, let us compare the two. What differences can you see? Yes, the increase in the volumes has been enormous. So, these raisins which were super dry have absorbed a lot of water and have increased in volume enormously. This is imbibition. Moving on, imbibition is also diffusion since water movement is along a concentration gradient and the seeds and other such material have almost no water, hence they absorb water very quickly. Now, what is important? that water potential gradient should be there, otherwise imbibition will not take place. And in addition, affinity between adsorbent and the liquid is also required for imbibition to take place. Different type of organic substances have different imbibing capacities. Proteins have high imbibing capacities as compared to starch, and that is why proteinaceous pea seeds swell more or imbibition. Now, when imbibition occurs, there is an imbibition pressure also because of the increasing volume of the imbibant. This pressure can be of tremendous magnitude. You know, in earlier times, people used dry woods soaked in water to crack large rocks. The imbibition pressure would be so high that the rocks would be cracked just by putting a piece of super dry log and then soaking it in water. The people were able to break big rocks. Even imbibition pressure is very high for the little seedling that is germinating. The seed when germinates, how does it germinate? It absorbs, it imbibes a lot of water, and as a result a pressure is built up, and that is how the young seedling is able to come out of the seed. Otherwise, how can you imagine will it be able to come out of all the soil and all the weight that is put over the seed? You know, we sow the seeds deep down, and then the young seedling, it needs to come out, removing all the soil that is over it. This happens because of imbibition pressure. So, we can compare the osmosis to imbibition also. While one is movement of water from high concentration to low concentration, but here the solvent is being absorbed by solid particles. Osmosis requires a semi-permeable membrane, but no such requirement is there for imbibition, and the pressure is osmotic pressure. It is not very high. The pressure that builds up during osmosis, it is not a very high pressure. On the other hand, the imbibition pressure, it is very high comparatively. Also, during osmosis no heat is produced, but imbibition produces heat. And lastly, osmosis is dependent on the presence of solute particles, but it is dependent on the presence of colloidal particles. This was about osmosis and imbibition. Now, let us move on to our practice of some questions that have been asked in competitive exams in earlier years. Let us have a look on few of them. Suppose a leaf peeling of Triscantia is kept in a medium having 10% salt solution. After a few minutes, if we observe the leaf peel under the microscope, what will we see? Obviously, we will see that the water has exited from the cell, that is the plasmolysis has occurred. Next question. When dried seed of pea are placed in tin and water is added up to their level and then a lid is put lightly over it, within an hour the lid will be blown off. This is the assertion, and the reason is due to rapid cell division in pea seeds. So, they are saying that the seeds have increased in size because of the rapid cell division. You have to tell what is correct. Obviously, we have discussed just now that assertion is true. The pea seeds will increase in size, but the reason is false. They will not increase because of cell division. They will increase in size because of osmosis. Very good. Next, the process by which water enters into the seed coat when a seed is placed in suitable environment for germination is again imbibition. Again, we are comparing the pea seeds and wheat grains. They are soaked in water. The pea seeds showed more swelling than water, and the reason? Oh, I have just now discussed it. If you had listened properly, you must be knowing the answer, that the reason is imbibition capacity of proteins is much more than that of starch. That is why the pea seeds, they get bigger in size as compared to wheat seeds, which have mostly starch. Okay. Now, there are some questions for you to try on your own, and please try to solve them all. If water enters in a cell, the pressure exerted by its swollen protoplast is osmotic diffusion pressure deficit, turgor pressure or imbibition. Living cells placed in isotonic solution, that is 0.9% NaCl solution, retain their size and shape. This fact is based on the concept of facilitated diffusion, osmosis or transpiration. Now, in this the values of osmotic potential and pressure potential of 4 cells have been given, and you have to identify the correct sequence that shows the path of movement of water from among the following. So, calculate and find out the path of movement of water. This was all about today's class. We will meet in next class to discuss these answers and study about the transpiration pool. Thank you.